Okay, let's talk about some things that we might find outside of prokaryotic cell walls. So when we take a look at a prokaryotic cell, like a bacterium, the inside is a little bit simplistic. You'll have a circular chromosome, you'll have ribosomes, you'll have enzymes, you'll have cytoplasm, but there are not membrane-bound organelles like you would find in a eukaryotic cell. However, the outside of the bacterial cell can be really complicated and complex. So we're going to take a look at some of the things we might find coating the bacterial cell wall. So it's important to understand that all cells use a plasma or cell membrane as their boundary. And in this picture of a prokaryotic cell that I have here, I've basically taken a look at a box and we're going to redraw what we see in that box. So what we have in that box is going to be a plasma or cell membrane, and some sort of cell wall on the outside of that. So the plasma membrane and the cell wall are really important components to bacteria, and we're going to be covering those separately in a different lecture. What I am interested in today is what is outside of the cell wall, if anything. So in other words, we're going to be taking a look at what might be beyond the cell wall that's still attached to the bacteria. So again, I just want to make sure that you understand we are not looking at the cell wall or the plasma membrane today. We are looking at things that we might find outside of the cell wall. So the first thing that you might find outside of the cell wall is some sort of protrusion or an extremity of sorts. So for instance, we might have something that sticks out like this, or we might have a little tail. These are all things that might be sticking outside, protrusions. So let's go ahead and cover those. So the first thing to note is that when we look at protrusions or extremities, there are really three different types that we might find. The first is flagella or flagellum. A flagellum is singular, flagella is plural. And flagella are used to help move. So flagella or flagellated bacteria are going to move the bacteria. Now, not all bacteria are flagellated, but the ones that are capable of effectively swimming. And so if they become pathogenic, they are able to swim away from our white blood cells, which can be problematic to us. So when we think about flagella, let's look at how they might be anchored. So this image shows how flagella are anchored into the plasma membrane and the cell wall. So you can see that at least one part is anchored into the cell membrane or the plasma membrane, and then the flagella sticks outside of the cell. Now, when we think of flagella, most of us think of sperm, which has a single flagellum, but that's not always the way that bacteria do it. So what I think is really interesting is that, yes, we can have that single flagellum, like we see in this image here, or we can have a whole bunch of them at one side, like this one, that almost looks like a kraken, or we can have flagella at opposite ends, or we can actually have flagella that kind of like stick out everywhere all over the cell, interestingly enough. So it's not always a single flagellum located at one end of the cell, but rather we can have a variety of different designs. And again, I want to remind you that not all bacteria are able to make flagella. And the ones that are able to make flagella are often able to what's called upregulate or downregulate their flagella. In other words, they're able to make their flagella and put it out so they can swim away, or take it away so that they don't swim anymore. Now, flagella are useful for movement, and not just any movement, but what is called directed movement. So let's go ahead and talk about that a little bit. So flagella allow for directed movement. Now, what would they be moving towards or away from? If they are moving towards or away from a chemical of some sort, like sugar, or away from a toxin, that's also a chemical. We call that chemotaxis. So the term taxis is like a taxi, which means movement, and chemo refers to chemical. So movement towards or away from a chemical. Again, it would make sense that they would move towards like a sugar or something yummy and away from something like a toxin. You can also have phototaxis, which is movement towards or away from light, depending on your preference. If you're a photosynthetic organism, like the euglena that you may have seen from Lab 5, then you would want to move towards the light because the light gives you energy. However, if you're photosensitive, you might move away from the light because you don't want to be near the light. So directed movement, which is taxis, is just going to be called chemotaxis if it's in response to a chemical, or phototaxis if it's in response to light. Now, how do they use these flagella to move? Interestingly enough, the way that bacteria move is through what feels like a game of hot and cold. So what do I mean by that? Well, let's say that I have a room. And this is going to be a top-down picture of a room. And I've hidden a prize right here that you can't see. And we're going to play hot and cold until you find the prize. And you start over here. Okay, you have no idea where the prize is. So you start moving this way, and I say warmer, 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 colder, and you go, oh, nope, colder, colder. Okay, warmer, 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 really hot. Okay, and you go, oh, colder, cold, nope, warmer, warmer, and you eventually get there. So in other words, you move towards it in kind of this zigzag line, playing hot and cold. Well, bacteria can't see, but they can sense. Let's apply this game to the bacteria that's trying to find sugar. In this image here, I have a bunch of purple dots, which represent sugar molecules. And now I have my bacterium that's just kind of moving along. And that bacterium is going to run into, just by random chance, this sugar molecule. And they have a sensor, a receptor on their surface, and they go, oh, we just ran into sugar. I wonder if there's more. And so they start moving. And so they play this hot and cold game where they do directed movements towards an area where they think there's more. And so like in this area that I've moved that bacterium into, it's now into an area where there's more sugar, right? But if it starts moving this way, it's going to run away from the area that has more sugar. So it's actually going to do this combination of what's called a run and tumble, which is basically hot and cold respectively, until it hits the jackpot. So let's say it runs in that direction and it's getting closer and getting closer and getting closer, and then it kind of reorients itself with a tumble. Like I'm starting to run away, so let's see if I can find it. And then I'm going to run this way. It's just random. It doesn't know which way to go. And it realizes that when it does that, it's moving too far away, so it's going to tumble again. And it's going to try to reorient itself up. Nope, even farther. I'm going to tumble again. And I'm going to change direction. I'm going to go this way. And you can see it's like getting closer, now farther away. And now we're going to tumble again and reorient ourselves. And up, farther and farther away, we'll tumble again. And eventually, through this game, it finally hits the jackpot. So it does these series of runs and tumbles. Now, when it does a run, the flagella move one way and kind of power it forward in a really fast zoom straight forward. When it tumbles, the flagella actually rotates the other way and it kind of vibrates like, ugh. And really all it's doing is it's reorienting itself in solution. so that it can get a new orientation, and then it will try another run. And depending on what it senses, it will reorient itself again for another tumble, etc. And it will go through this process until it gets to the jackpot. If that chemical were a toxin, then you would see something like that away from the toxin rather than towards it. But that same process. So directed movement with flagella is achieved by using a combination of runs and tumbles. And runs tend to be pretty fast directed, and tumbles are kind of just like rotate around and reposition. Let's go ahead and watch a video about how this happens. This video shows bacteria doing tumbles and runs. The center bacteria is tumbling. The bacteria that are moving across the screen really quickly are doing runs. So you can see just how fast it is. And the ones that are just sitting there, oh, there's kind of a funky little one coming by, using a different form of motion. But again, you can see that the tumbles are about reorienting themselves. They just kind of vibrate around, and then the runs are directed and fast. Okay, now that we've taken a look at that video, now we can see this, and we can see that when we have a run, the flagella goes one way, and when we have a tumble, the flagella goes the other way. In my opinion, it's probably the easiest to see in this one, where you have one of those bacteria that have a lot of flagella kind of spread around the cell. And so when they get ready for a run, all the flagella kind of like band together towards the back and act like a propeller, like shooting it forward. And then it unravels and they all spread out, and it's kind of like, oh, as they tumble around. You do not need to know counterclockwise or clockwise. What I do want you to know is that directed movement is always a combination of runs and tumbles. To be able to do that, you need a flagella, at least one, and that you'll rotate your flagella different ways to achieve a run or a tumble. And as I said earlier, you've got this game of hot and cold, and so you get this like combination of runs and tumbles to get towards where you want to go. All right, so that's it for flagella. Now let's come back up here and take a look at some other protrusions that we might encounter. So again, these are all optional, not all bacteria have these. But another protrusion that we might encounter is called a conjugation pilus. Now, a conjugation pilus is sometimes also known as a sex pilus, which is misleading because it suggests that bacteria have sex and they don't. I don't know why. Well, I have ideas why, but I don't actually know why they decided to use this term like male and female bacteria and sex pilus, because those are not real things. Perhaps they just tried to kind of make it more like the animal kingdom so that it would be interesting to students, so that it would be less boring, because if somebody says sex pilus and then shows you a long protrusion, everybody's suddenly wrapped with attention, like what's going on? And when you say conjugation pilus, people are like, oh, I'm tired. Okay, I don't know, but we're going to use the real term, which is a conjugation pilus, because that's what it does. So let's take a look at how this works. A conjugation pilus is going to be a sky bridge that is synthesized by one bacterium, and it basically extends out and joins to another bacterium. So here you have one bacterium that is called the donor, and then they basically extend out this conjugation pilus to the recipient. And in my opinion, that's like a tunnel between the two cells. And the tunnel allows for exchange. Exchange of what? DNA. So a conjugation pilus allows for exchange of DNA between one cell and another. Okay, we will learn more about this particular process of sending DNA from one bacterium to another when we get into topics 5 and 6. The third and final protrusion that we might encounter is called fimbria. Fimbria are little hair-like spikes that stick out of bacteria, and they act like little gecko fingers. So what do fimbria do? They allow for that bacterium to attach to some other surface, like let's say an implanted piece of metal, or your own tissue, or even other bacteria, but typically it's used to attach to your tissues. Okay, going back to this picture, you can see that there are fimbria here. They're sticking out. So all these little what looks like sharp spikes, those are actually hair-like appendages that are called fimbria that are sticking out and allow that cell to attach. Here's another picture of bacteria that are expressing fimbria. And once again, this is optional. Not all bacteria are going to make fimbria. So there's more fimbria. But those that do make fimbria, that actually helps them establish a presence in your body. So fimbria are important pathogenically because they actually contribute to disease. So let me give you an example. One of the most common types of infections is called a urinary tract infection. A urinary tract infection gets the abbreviation UTI, urinary tract infection. UTIs are usually caused by bacteria that crawl up from the urethra into the bladder. To prevent urinary tract infections, one of the best defensive mechanisms is frequent urination. So why would frequent urination be helpful in preventing urinary tract infections? Well, if I take a look at a bladder here, so this is going to be a cross-section of a bladder, and then here's the urethra, and then here's the opening. So when you get, urine is stored in the bladder, like so. And then when urine gets ready to come out, and when that happens, you force a lot of fluid into a small hole, the fluid shoots through at a really high velocity. So this is why when you're peeing, the pee comes out pretty quickly and with a fair amount of force rather than just dribbles out of you kind of slowly. Well, the reason why that's good in terms of preventing urinary tract infection is because if you have any bacteria that are like crawling up your urethra trying to get into your bladder, that water, that urine, should knock them off. It should feel like Niagara Falls, like someone clinging to a rock in Niagara Falls, and then just the sheer force of the water knocks them off and into the toilet, where they can no longer crawl up into your bladder and cause an infection. But some strains of E. coli produce very robust fimbria, and when they do, they can hang on and attach to that wall even when the urine is streaming past really quickly. And so they hold on for dear life and they're like, bye-bye, and then they keep crawling up and they're able to finally get up here. And once they're in the bladder, you have a bladder infection, which is a type of urinary tract infection. So While we say that frequent urination is the best prevention to prevent urinary tract infection, you may encounter patients who get repeated urinary tract infection despite drinking a lot of water and urinating frequently. And it's possible that they are afflicted or infected with a type of E. coli that just has a really strong fimbria, and that even frequent urination doesn't dislodge all of the bacteria from the urethra. So that's a case in point about how Fimbria can contribute to a bacteria's ability to cause disease. Okay, so now that we have finished looking at protrusions, let's take a look at some external layers. So what do I mean by an external layer? What I mean is some other layer that is more coating, not a protrusion per se, but another layer outside of the cell wall. So do we have an additional layer? And again, these are optional. Many bacteria don't make additional layers beyond their cell wall, and the ones that do don't always make them available. So you can be a bacterium that has the capacity to make an external layer, but that doesn't mean that you do all the time. So let's take a look at what some of those options are. Okay, one possible outside layer is referred to as a surface layer, or abbreviated an S layer. An S layer is a shell of repeating proteins. S layers tend to be very symmetrical looking when you look at them under an electron microscope. And they have a number of advantages for the bacteria that make them. One advantage is that it makes it harder for our immune system to find them and to capture them. Now, understanding why this is true requires a little bit of knowledge about how the immune system works. But effectively, the immune system is going to look for things on the surface of any pathogen that are unique and provide good opportunities to grip. So if I show you a picture of an S layer, you can see that it looks really symmetrical. In other words, there's not a lot of places to grip and there's not a lot of differences. So if you don't recognize whatever one of these little guys are, then that is basically invisible to your immune system. The other advantage that they confer is that they allow for the bacteria to withstand a greater change in pH. So why is that? Well, proteins are good buffers, and because they are good buffers, they can actually suck up some of the extra H plus or give off extra H plus as the pH shifts. So you're going to learn about buffers in chapter 3 of your workbook, but if you haven't gotten there yet, I'll real briefly talk about it. So if I have an acidic environment, for instance, an acidic environment is going to be one where there's lots of H pluses. And those H pluses can what's called denature, and you're going to learn about that too, other protein in the bacteria. In fact, that's what would be toxic about an acidic environment to bacteria, is that it actually unravels their other proteins. So what the S layer does is the S layer is almost like a bunch of expendable proteins that will become unraveled instead. So if this is your S layer protein, this is just one protein from the S layer. Remember, the entire S layer is going to be a whole series of these, like so. Then what will happen is they will actually suck up those extra H pluses. And of course, they'll be unraveled a little bit, but they were expendable. And so by doing so, they remove the H pluses from the solution so that it doesn't actually attack the real necessary proteins. So in this way, the S layer provides some expendable proteins that can help suck up additional H pluses or give off additional H pluses and protect the more internal but necessary proteins that the bacteria needs for survival. Okay, so that's it for the S layers. The last layer that I want to talk about is called glycocalyx. Now, like the S layer, not all bacteria can make glycocalyx, only some can. Glycocalyx is made of carbohydrates, it's made of sugar. So if you think about glyco, that usually means sugar. So glycocalyx is made of carbohydrates. And there are two ways to spin this carbohydrate or sugar web outside of your cell. If you can make glycocalyx, but you basically spew out these carbohydrate strings in kind of a loose aggregation, that's called a slime layer. So there's two variants here. But if you wrap it up tightly around your cell, that's called a capsule. So I want to talk about the slime layer first. When I was a kid, I used to love watching cotton candy machines. And if you've ever been to a fairground, for instance, maybe you've had that opportunity to watch a cotton candy machine because it's really fascinating how it works. So you get these sugars that basically end up in this machine, and I really don't know how it works, but what gets spewed out of this granulated sugar are like these spider webby pieces of sugar, right? I mean, we know that cotton candy is in fact like sugar that's almost been fluffed, like pillow fluff, and it kind of spews out around this container, and then you use one of those wands, those little cardboard wands, and you kind of pick up the sugars. And so when I think of slime layers, I think of bacteria acting like a cotton candy machine. So you'll have a bacterium that is capable of producing a slime layer. And the slime layer kind of gets shot out in this kind of like spider web like aggregation. That may or may not be kind of attached to the cell, but it just gets spewed out here like that. That slime layer, which is a lot like cotton candy, sticky like cotton candy, is going to be a welcome mat for other bacteria to land and attach to. And we see slime layers with plaque. So plaque is going to be some bacteria laying out slime layers and then other bacteria attaching to that slime layer and potentially getting coated in that slime layer so that our immune system can't find it. So if it is loosely spewed out like cotton candy, that's going to be a slime layer. Now, the other option is that the bacteria doesn't like spray it out everywhere as a welcome mat for other bacteria, but rather puts it out and wraps it tightly around itself, more like a spider wrapping up an insect in a cocoon. And if that's the case, we call that a capsule. So if this bacterium was instead to secrete glycocalyx as a slime layer, it would look more like this. And so you can see I'm keeping it really close to the cell that's secreting it. So it's wrapping up like a cocoon. So slime layers and capsules serve different functions. Slime layers are there more for attachment, like to attach to teeth, where capsules are for protection, mostly against our immune system. So our immune system has cells that go around and eat stuff up. And since I'm an 80s child, I think of our immune cells like Pac-Man. Now, not all of our immune cells do this, but we do have a set of immune cells that do go around and basically eat away your problems. But part of being able to eat away your problem is being able to get a grip on your problem so that you can ingest it. And encapsulated bacteria are like slippery fish. If you've ever tried to hold a live fish before, they have a little bit of slime on their surface, and when you try to hold them, they kind of wiggle, 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 and they can get out of your grip really easily. And so imagine if you were, for instance, a bear that was salmon fishing in a river, but you didn't have talons, so you couldn't pierce them. And you just had to use like the pads of your paw to try to grab a salmon and eat it. You would really struggle to get enough food because you would constantly be trying to pick up that fish, and the fish would keep getting out of your grip. So in the same way, the capsule is like that layer, that slimy protective layer that makes it really difficult for our immune cells to try to get around it and eat it. So capsules help bacteria infect our bodies. Now let's take a quick look at this. So again, this just shows you what I talked about. Glycocalyx is going to be that carbohydrate kind of stringy stuff that you secrete out. And if it's loose, it's a slime layer. If it's tight, it's a capsule. Now, encapsulated bacteria don't always produce their capsules because it's really energetically expensive to do so. In this image here on the right, the actual bacterial cells, I'm going to highlight them in pink here, are the black spots. That's the volume of their cells. That green stuff, that's all capsule. Look at the volume difference. If you were a bacteria cell and you tried to make a capsule, basically you would be looking at making something like really big around you. So if this is you, and you have to make a capsule, you're going to have to eat enough food to be able to secrete that much substance around you. Wow, that's a lot of food you're going to have to eat. So what this means is that it's very expensive for bacteria to make a capsule. So they don't usually do it unless they're in trouble. Okay, that concludes this lecture that looks at different types of external layers and protrusions outside of the cell wall. Thanks.